introduction, Rissa. So my name's Chris Basting. I am a researcher in Dr. Nichols Class Lab in the Department of Surgery. I'm also a PhD student in the biocrates program here. And so today I'm going to be talking a little bit about our projects with biocrates and CMSP, specifically investigating stool and plasma metabolome of people with HIV. So in our lab, we primarily look at the intersection of systemic inflammation and mucosal dysfunction, microbial dysbiosis, and immunity and inflammatory responses, and how they relate to several different contexts, including women's health, COVID-19, and other infectious diseases like HIV. So we generate a lot of microbiome data, analyze a lot of microbiome samples. And so the microbiome is a collection of microorganisms in the environment. You have microbiomes, multiple different body sites, including gut, which is one of the largest ones, as well as vagina and skin. And so these microbiomes are, they consist of all the bacteria, viruses, fungi, that, are in that location. It also consists of all the genes and the functional, content that they, have inside. So the concept of dysbiosis is basically an imbalanced microbial community. And this can happen through drugs such as antibiotic usage or infection, which disrupts kind of the natural microbiome. And that can result in changes in the abundance of microbes or the change in their function. So in the microbiome, we know it's critical for health, but it's regulated by a lot of factors, which makes it kind of difficult to study sometimes. It's influenced by genetics, environment, diet is a big influencer. Lifestyle, like sexual preferences can largely influence your microbiome, and other things like, medication maybe that you're taking. But the microbiome has a lot of important functions as well in, the sites that it's located in. The gut is, probably most well known for it, the microbiome being very important in, digestion of food and getting all the nutrients that you need. And then in the vagina, the bacteria there are very important in maintaining a low pH, which is optimal for things like preventing STIs. And kind of the way that the microbiome influences these effects is through the production of metabolites that they produce. And so the metabolites they produce can have a significant effect locally on the tissues that they're close to, but it can also have systemic effects. Once these metabolites can cross the mucosal barrier that they're in and into systemic circulation. So some of the primary metabolites the microbiome produces are going to be short-chain fatty acids. These are going to be produced by the fermentation of dietary fiber. You have secondary bile acids, which are produced by conjugation of primary bile acids that your body kind of releases into the GI tract. And then amino acid metabolites, what are going to be derived mainly from your diet. So tryptophan is a major amino acid, and how tryptophan gets metabolized by the microbiome into the microbiome metabolizes tryptophan into many different derivatives. So, kind of you can see on this plot that, these metabolites have influences on many different functions, across the entire body. Lipid oxidation, insulin sensitivity, blood pressure, they are having influences all across your body. And so differences in the composition of the microbiome or diet can impact the abundance of these metabolites and thus potentially some, health outcomes or stuff like that. And so microbial dysbiosis is associated with many diseases. In our lab, we primarily focus on COVID-19 and HIV, but it is associated with many diseases. You can probably find a paper associated with microbiome with about any disease you could think of. But we're primarily interested in COVID-19 and HIV, and those infections disrupt the microbiome across, kind of multiple different sites. So COVID impacts the oral microbiome, lung microbiome, the gut microbiome, HIV, primarily the gut microbiome, vaginal microbiome. So these are the sites that we look at when studying. In terms of HIV, so there is still no cure for HIV. This figure on the left basically shows the potential survival gains in terms of years for a person infected with HIV throughout the course of time. So in the early 90s, people who were on antiretroviral therapy when it was still kind of new only had a expected life expectancy of about 28 years. So pretty small gains in terms of survival with ART or antiviral therapy at that time. As those drugs have gotten better, the life expectancy has increased a lot, improved a lot, close to what it is for an HIV negative individual, but it's still not quite the same. And this is largely because people with HIV have chronic inflammation and immune activation that persists even when they're on these drugs. One of the reasons that HIV is so hard to cure is that it is a retrovirus that kind of infects long-living CD4 T cells. So when you're infected with HIV, you have a lot of virus in your system. When you take ART, it quickly drops to undetectable. But if you ever stop taking those drugs, the virus will rebound in those long-living CD4 T cells and begin replicating again. So to effectively cure it, you need to clear all of the latently infected cells. And that's essentially what's called the virus, the HIV virus. And as an impact, so, a lot of people, I think especially in the States, think that HIV isn't really a problem anymore. The drugs are so good. But in 2023, there's an estimated about 40 million people living with HIV globally and about 1.3 million new infections. So these are people that, still living with HIV as a chronic condition, and they're at higher risk of a lot of comorbidities like cardiovascular disease that can have impact on quality of life. One of the causes of the chronic inflammation that exists in people with HIV is microbial dysbiosis and mucosal dysfunction. So on the left, we have a healthy gut. In a healthy gut, you have a protective layer of mucus that prevents microbes from interacting directly with the intestinal barrier. You also have an intact epithelial barrier. And you have a healthy microbial community, which in general, a gut is characterized by high diversity. You also have kind of homeostatic interactions between immune cells in the lamina propria and microbiome that kind of work together in a back and forth to help keep things regulated and keep everybody happy. In an HIV infected gut, so HIV infects CD4 T cells, and the gut is actually the largest reservoir of CD4 T cells in the body. So upon HIV infection, a lot of these cells are killed off, and a lot of them are regulatory cells that their job is to maintain that homeostasis. So what ends up happening is the epithelial barrier breaks down. You have translocation of microbes and their products into the lamina propria, and that kind of activates your immune system and causes additional inflammation that causes systemic inflammation across your body. You also have an altered microbiome in HIV infected people. It's kind of complicated because it's impacted by a lot of different things like diet and lifestyle that are specific to the population of people with HIV, but generally you have to reduce diet. So we're really, kind of interested in microbial mechanisms that are contributing to chronic inflammation and immune dysfunction. And so with biocrates and metabolomics, we're basically asking, how the metabolome differs in stool and plasma between people without HIV and people with HIV. Within people with HIV, what are the associations between the metabolome and markers of inflammation and immune activation? And then how can we utilize microbial metabolites to mitigate chronic inflammation in people with HIV? So just an overview of some of the projects that we've done. So far, we've primarily done kind of smaller pilot projects using biocrates, using the MXT Quad 500 kit. So we've looked at the plasma of HIV positive and HIV negative individuals, the stool of HIV positive and negative individuals, and then we've also tried looking at the stool of HIV positive individuals using these DNA Genotech ME200 collection kits, which are nice because they're stable to metabolome for about 7 days at room temperature. So we've used them to give to our patients so that they can do an at-home collection and then mail it over a lab, which makes sample collection a lot easier. And then all the individuals that have HIV in these studies are all virally suppressed. So getting into some of the data, this is looking at the plasma metabolome of HIV negative and HIV positive individuals. This is another principal component in all of this, where each individual represents, or each point represents a single individual's basically entire metabolism as measured by biocrates. And the distance between individuals represents how similar they are, with, points that are more close together are similar metabolomes, points that are more further apart have different metabolomes. Essentially what we see in the PCA here is that there seems to be some clustering of, the metabolome by HIV positive or HIV negative status, which is the colors here. But there's still quite a bit of significant overlap between the groups. So, maybe as a whole, the metabolome of these individuals is not, maybe super different. But when we look at when we do univariate analyses of the individual metabolites, we start to see kind of some interesting differences. And that's shown in this volcano plot, where we have elevated metabolites in HIV positive individuals on the left, and HIV or metabolites elevated in HIV negative on the right. And I'll go into these further detail. So in the plasma, one of the interesting variables that we saw was PLA2 activity. This is one of the metabolite indicator variables that James is talking about. So a very useful kind of variable, and there's actually several variables of PLA activity in the biocrates kit. There's like several ones to kind of measure it. But it's basically the ratio of phosphatidylcholines to the ratio of isophosphatidylcholines, and that's an estimate of the activity of this phospholipase K2 enzyme. So it's been shown that PLA2 activity is associated with increased inflammation, associated with increased risk of cardiovascular disease. So the fact that it's elevated in people with HIV potentially makes it a good biomarker for us to evaluate, a mechanism that these individuals have for increased cardiovascular disease. And, if it essentially these fatty acids can cause plaque formation. So if we can potentially address, the reasoning behind why these lipids are altered in people with HIV, it could be a mechanism to reduce cardiovascular disease in people with HIV. We also see decreased carnitine in plasma of HIV positive individuals. So carnitine plays an important role in mitochondrial energy metabolism. It basically shuttles long-chain fatty acids into the mitochondria that the mitochondria then uses to get energy. So when you have decreased carnitine or deficiency in carnitine, it kind of makes your mitochondria more dysfunctional, and that in turn makes your cells more dysfunctional because they don't get the energy that they need. And interestingly, carnitine deficiency is more common in people with HIV, and mitochondrial dysfunction is like a hallmark of HIV infection. And that's associated with all sorts of negative effects like accelerated aging, cardiovascular disease again, and just like fatigue in general. So, for us, this is another potential variable. Like if we can address the reasoning behind this decreased carnitine, that might be a way to address mitochondrial dysfunction in people with HIV. Looking now at the stool, we actually see kind of larger differences between HIV negative and HIV positive stool metabolome. It's kind of shown by the better clustering, at least in the heat map, between HIV positive and HIV negative. We also see kind of larger increases or differences in some of the metabolites in the stool. Same kind of volcano plot with metabolites increase in HIV on the left and increase in HIV negative on the right. And I'll go into more detail on these in the next slide, but some of the ones that stood out here, at least the triglycerides. So it's nice, at least with biocrates, that it provides specific triglycerides that you can do analysis on or look up in literature. But altered lipid profiles are common in people with HIV, especially high triglycerides. So we see some of the actual triglycerides being elevated in some of these HIV individuals. We also see that diglycerides are actually elevated in people without HIV. This is something that I haven't really seen a whole lot in literature, and I don't really know exactly currently what the clinical implications of that would be, but it would certainly be an interesting thing for us to follow. So just more looking at the stool metabolome, one thing that we did see was decreased tryptophan metabolites, or 3-indole-propionic acid. So this is a tryptophan metabolite that's produced by the gut microbiome, specifically Clostridium species. And this metabolite helps maintain mucosal homeostasis and barrier function. It's also associated with lower diversity in the gut. So, reduced concentrations in HIV individuals could reflect the fact that they have reduced diversity in their guts. Could also reflect, differences in the microbiome, that maybe there's higher abundances of bacteria that lack the ability to produce this metabolite. So if we can, address or change the microbiome of HIV positive individuals to maybe produce more of this free IPA, it could maybe be a possible mechanism that we can improve barrier function and mucosal homeostasis in people with HIV. Switching gears a little bit now to looking at just within people with HIV. So I mentioned this a little bit, but so upon HIV infection, it infects your CD4 T cells. And so after infection, your CD4 T cell count drops rapidly, in the bottom left figure. And your CD8 T cell count increases rapidly to try to fight off the infection, and your CD4 T cells are dying. But once you start antiretroviral therapy, ideally you want that kind of ratio of T cells to invert, where your CD4 T cells are, increasing and your CD8 T cells are then decreasing in. But only a portion of individuals with HIV have this actually happen to them. And although CD4 CD8 ratio, while you're on Earth, is correlated with inflammation, immunosenescence, and increased risk of comorbidities and mortality. There's one paper that showed for every 10% increase in your CD4 CD8 ratio, you have a 15% decrease in your risk for mortality. So in general, we'd like to use this ratio to determine like immunological outcomes for people with HIV. So now looking at these DNA genotype ME200 samples that we collected, these are all in people with HIV. And we saw that there was a positive association between lactic acid in the stool and the CD4 CD8 ratio. So lactic acid is produced by a lot of gut bacteria, primarily lactobacillus species or bifidobacterium species. These are also very common in a lot of probiotics that you'll get over the counter. And lactic acid helps maintain epithelial barrier integrity. It also regulates the microbiome by preventing potentially pathogenic bacteria from overtaking the microbiome. And it can regulate immune cells, and that's kind of a newer thing that's coming out, is the ability of lactic acid to regulate the immune system. But the fact that we see a positive association between lactic acid and CD4 CD8 ratios implies to us that there is something important going on with lactic acid that's maybe improving immunological outcomes in people with HIV. And interestingly, we can measure the HIV viral reservoir in these individuals, or we did measure the viral reservoir in these individuals, by essentially measuring the number of virally positive cells that were in their lymph nodes from a lymph node biopsy. And we see a negative correlation between lactic acid concentrations in stool with viral RNA in the lymph nodes. So this is kind of a potential mechanism, that lactic acid maybe has, some effect on the viral reservoir, and that could also explain, by decreasing the viral reservoir in people with HIV, that might be a reason for it to be associated with good immunological outcomes. And also what's cool is that, in the last probably 4 years or so, there's been several publications that are talking about the importance of lactic acid in people with HIV. It's been shown to promote CD4 T cell recovery in people with HIV. It's actually been shown to have inhibitory effects on HIV replication. And it's been shown to induce kind of anti-inflammatory responses in macrophages by kind of epigenetic means. So, and then there's also been a clinical trial, it was 2 years ago, where they gave people with HIV that had low CD4 CD8 ratios a lactic acid bacteria probiotic, and saw small increases in their CD4 CD8 ratio with this probiotic of lactic acid bacteria. So this just is a good indicator that lactic acid could be a good microbial metabolite to improve HIV outcomes. So in conclusion, we see increased biomarkers of inflammation and mitochondrial deficiency in people with HIV in the plasma. We see altered tryptophan metabolites, specifically 3-IPA in HIV positive stool. lactic acid concentrations in stool were associated with good immunological outcomes, and increasing the lactic acid production by the gut microbiome through maybe diet or probiotics to help mitigate chronic inflammation in people with HIV. And then lastly, just metabolomics and biocrates in general has allowed us to better understand the function of the microbiome and how it relates to HIV infection. Like others have said, it's really nice to have actual names for all your metabolites. The metabolome indicator variables have been really helpful. And then future things for us to do would be like a pathway analysis, as well as we have the matching data for microbiome in these samples. So correlating metabolites from the biocrates kits that we did get with the abundance of microbes in stool can help us maybe identify specific bacteria that are, maybe important in these metabolites. So, with that, I'd just like to thank my lab, and especially Candace and Marissa, for organizing. So, thanks. Take the question.